Welcome. I think we're going to go ahead and get started. It is a pleasure having all of you here today. This is a very exciting day and one that we have been looking forward to for quite some time. I want to welcome everybody who's attending today, and this is going to be a webinar about stem cells, and it's going to be a time that we have with Dr. Bharti to learn what stem cells are and how they potentially could be helping us with choroideremia treatments in the future, whether that be working in collaboration with other possibilities, other treatments, or solely by themselves. So I'm super pumped, and thank you again for joining us. We have a couple of just housekeeping things to go through first. And as most of you who have been here before know, we're using Zoom and we're using the webinar feature, which means all of your great questions can be asked, but use the chat feature. And I'll be sure to forward them on to Kapil either at in the middle or at the end of our session. I'll make sure we get them covered today. I would be remiss without saying a thank you to our sponsors. We received some funding a while back ago from Allergan and Biogen. So we always want to make sure we put that out as a very generous thank you to them as well. So those are the two housekeeping issues. And I just want to say, hi, Kapil, Dr. Bharti. We've met with him several times, Eric and I at the NIH. He has joined us at our symposium, science symposium, and he is a great friend to our organization. And I love that you're wearing your shirt. Thank you so much. Woo! He is, I just need to say, he is a very special person. He works at the NIH, National Institute of Health, and where you are a senior investigator with stem cell and ocular cell research. He sits on our science advisory board, and that means he has an important role with our organization as he directs and he influences and he shares his thoughts on research that we're doing in trying to fulfill our mission of the Choroideremia Research Foundation, which is trying to find a treatment, ultimately a cure for CHM. So we're super pumped, as I said, to have you here, and welcome aboard. And I will turn off my stuff so you can take over and start giving us your great information today.
Thank you, Beth, and I want to thank the foundation for inviting me to talk about our work and about stem cells. One announcement from my side, as many of you know, or all of you know, that I'm giving this presentation from home because of COVID, and you may have already heard. If not, you might hear during the talk our new puppy. She's very excited to show her support from time to time, so please excuse that. With that, I'll get into the discussion for today's topic, which is around how stem cells can be used to develop a cure for choroideremia. And I say can be is because this is work in progress, and I'll show some of the efforts that we and others are doing. But we haven't reached a stage where we are ready to transplant stem cell-derived therapies in choroideremia patients. However, we're doing this work in age-related macular degeneration patients, and I think that work provides a big input into the choroideremia work that we have started. So as, again, most of you are aware that patients with choroideremia start off often with night blindness, and this is one of their common first symptoms. As the disease progresses, the loss of vision encroaches towards the middle of the retina. Often, in this case, although it shows middle part is clear, but they have spotty vision even in the middle. And that happens, these things are happening because the disease affects really the three main layers of the eye in the back of the eye, which is shown here on this slide. On the bottom here is you see a schematic of a human eye. The light is going through the cornea, the lens, and hits the retina where it's detected, and image is formed and sent to the brain where it's analyzed. These are the three main cell type or are important in image formation and visual input to the brain. On top, you see the photoreceptors that perceive the light actually and send the signals to the brain through other neurons. Next to the photoreceptors is the retinal pigment epithelium, which sits as a sandwich between the photoreceptors and the blood supply to the eye. And this layer has this very strategic location because this layer forms a barrier between the blood supply and the retina. and really controls how the nutrients go from the blood supply into the retina, and the metabolites go back from the photoreceptors into the retina, into the blood supply. In addition, the RP controls a lot of functioning of the photoreceptors. And for those reasons, if, as you can imagine, if the RP layer starts to dysfunction or dies off, very quickly photoreceptors start to die off and the choroid starts to die off. And in fact, that is something similar to what happens in choroideremia in advanced stages, where the mutation in the gene really affects the RP functioning initially and leads to RP atrophy, leading to a loss of choroid and eventually also the loss of photoreceptors and the retinal layers, which really leads to blindness in advanced stages. And as Beth mentioned also, really for advanced choroideremia, there is really no treatment at this stage. And the problem is you can't really have a treatment because the cell layers are gone, right? So the treatment would only work, some of the treatments, let's say drug or antibody or gene therapy, I'll come to that in a minute, would only work if the cell layers were viable. But here, as you can see, kind of really an example of this is a schematic example of what the back of the eye of a choroideremia patient would look like. The RP layer, as you can see, these cells with these hair-like processes, they're gone. The capillaries are gone in this part. You can see capillaries to the right, but here in the middle, they're gone. And as a result, above the lost RP layer, photoreceptors are gone. Eventually, the neurons that feed to the brain also start to die off. So all of these are the reasons why there's a significant vision loss in these patients. And once the cell types are gone, the only option we have is to replace them, and to replace them with quote unquote new cells. And so this is again making the point, driving the point home, is that as many of you are known, many of your friends and family members are participating in this gene therapy, ongoing gene therapy trials all over the country. Gene therapy is a way where you deliver. So for instance, in choroideremia, as you know, there's a mutation that leads to the disease. So a gene is not functioning properly, but in gene therapy, what you do is you add a new copy of the gene that is without the mutation and functions properly. And since this gene works in the RPE, in between the retina and the blood supply, all these attempts for gene therapy are to add that gene, new functioning gene now into RPE cells. But like I just said, if the RP layer is already gone, if the disease has already progressed to advanced stages, gene therapy is not going to work, because for that new healthy gene to express itself, it needs a healthy RP, it needs an RP layer, right? So really, gene therapy is only applicable to early to intermediate stages of AMD, of choroideremia, excuse me. But if the disease has progressed to really advanced stages, the only option we have is to now replace those damaged or lost cell types with stem cell derived cells. So here's a very simple way how we would make cells from stem cells. Eventually, I'll walk you through the entire process. Essentially, what we do is we start with blood cells of patients. We reprogram the blood cells into patients on stem cells. These are called induced pluripotent stem cells. They're called induced because they're from the patient itself. They've been induced to become stem cells. And then we can take these stem cells and make them into any cell type of the body, and in this case, for instance, RP cells. And I'll show the examples of making choroid and photoreceptors from these stem cells. And then the goal would be to now put the RP cells in a place where the RP cells are lost, right? So really in advanced stages, when the cells are gone, you want to put new cells in, made from patients on stem cells. And these RP cells, as like I said, the goal is to support photoreceptors and the choroid. As the new and healthy cells go in, they start replenishing the health and integrity of photoreceptors and the choroid, and would stop the disease from progressing further. So just again, going back to a little bit basics here, what are actually stem cells? Stem cell is a cell that has two main properties. One is that it can self-replicate, so that means it can make more copies of itself. So it can keep making more and more. So it's essentially an infinite resource once you have it. And the second important feature is that if you culture it under specific conditions, and I'll show you those conditions in a minute, it can make any cell type of the body. Essentially, this is how stem cells in an embryo make all of us, right? This is the stem cells, how they make the entire body. They can make blood cells, they can make neurons, they can make fat cells, they can muscle cells, any cell type of the body, these stem cells can make. Now, there are many different types of stem cells. I want to clarify what kind of stem cells we're using versus what are the other types of stem cells out there. So on top is embryonic stem cell, which I'm sure many of you have heard about because of their political contention around embryonic stem cells. These are the cells that are present in a developing embryo. These are the cells that make the entire body. They are so-called the true pluripotent stem cells because they can make all the cell types. We are not using these stem cells. We are using what is called induced pluripotent stem cells. These stem cells were discovered about 14 years ago. And what essentially researchers were able to do were to take any somatic cell, so like a little bit cells, blood cells from a patient, or a little bit skin from a patient, and reprogram those skin cells or blood cells into iPS cells, induced pluripotent stem cells, by adding certain factors that change the fate of these cells from differentiated back into stem cells. So they reverse them. Now, an interesting thing is that for all practical purposes, these iPS cells are almost identical to embryonic stem cells. And the beauty is that now we're no longer issues with political contention around these cells, and we can make them from patients on cells. And this would come advantageous when we go into transplantation, because once we want to transplant a tissue back into patient, if it's somebody else's tissue, patient's body would reject it. You may have heard about it from kidney transplants or liver transplants, that there's a big problem with transplant of what is called allo tissues from somebody else. Your body would immediately reject because they consider it as foreign tissue. But if they can be made from your own cells, then your body would not reject it. So there will be no need to give immunosuppression. So the long-term integration of these cells would be much higher. And then there's a third category that that is present throughout our adult life. These are called tissue-specific stem cells. Perfect example is blood stem cells. We know that our blood replenishes our red blood cells replenish every 3 months. So the cells in our bone marrow that keep making new blood, and the old blood keeps getting used up. So we're not going to talk about that. We will focus on iPS cells for today's discussion. Again, just to drive the point home, how are iPS cells made? Isolate any cell from the body. There's been many examples now: skin cells, fibroblasts, blood cells. People have even been able to isolate cells underneath the hair shaft and reprogram those cells by adding certain factors called the reprogramming factors. These are 4 proteins essentially that in about 3 to 4 weeks will give you pluripotent stem cells, iPS cells. And like I said, we can make blood cells, gut cells, cardiac cells, anything from those. And mind it, there are researchers around the world that are making all these different cell types for different disease indications for transplantation and other research. So what is the point? What is the goal that we are doing here? What is the project that we are doing here at NEI? Is that we start with patients' somatic tissue. We reprogram that into iPS cells. And in our case, we're interested in making the RP cells, the 4 receptors, and the choroid from these patients' iPS cells, and then try to put them together, either put them as a separate tissue into patient back as cell therapy, or together as a more complex tissue therapy that we're now currently working on. I want to put the expectations in a really realistic timeline, because these things take time. This is a very complex research. There's a lot of regulatory checkpoints that we have to meet to bring it to patients, and they're there for right reasons. And once they reach patients, again, there are further regulatory checkpoints before that therapy can be approved. And the entire process can take almost up to 10, 15, sometimes even longer, 20 years. The longest point, of course, is basic research or translational research, which I will talk about today, is because it takes a while for us to perfect the process of making. We're essentially recreating developmental biology in a dish, right? We're recreating, making a specific tissue, one specific tissue from stem cells in a dish, how it would have been made in an embryo, in an orphan embryo. So that takes time to perfect. And then to prove that what we made is exactly what we wanted to make, and it's safe, it's ready for transplantation, develop techniques for transplantation, and all of the work that the regulatory agencies like the FDA would ask us to do is needed at this stage. Once that is done, and I'll show you examples that a big chunk of this work has already been done, we get to FDA approval, where we start with very small phase 1 trials, again, to check feasibility of this process, whether work done in animals can actually be translated to human. And if it does in a phase 1, then we go to larger studies where we have more statistical power to show that the transplant indeed has the potential of treating or curing the disease. So it's a long process, requires a lot of patience and years of hard work. So now, sorry, just focusing on this part of the process. So preclinical work, what is called the IND enabling study. So IND is Investigational New Drug Application. That is a document that the FDA requires for us to put together to get an approval to transplant such cells or such tissues in patients. So an IND application requires that we demonstrate to the FDA that we can make these cells, not just in my lab, but in a special atmosphere, in a special environment called the clinical grade environment, which has a much higher standards of manufacturing because of control of, you can imagine dirt particles, viruses, bacteria. All of that is very tightly controlled, what's called the clean room manufacturing. That process takes a while, but we have to demonstrate that we can do that in the clean rooms, and we can do it reproducibly. And then what we make in the clean room, we again have to validate that it is true tissue that we intended to make. Once that's ready, we have to demonstrate that because we're transplanting cells into somebody's eye, we have to demonstrate that these cells, by transplanting them in animals, that they're not tumorigenic, they're not toxic, and they are going to do what they're supposed to do in the eye once they're transplanted. So we have to develop animal models to demonstrate all of these properties of our transplant. And then the tools that we use for transplantation or for making them, they're biocompatible, they're not going to cause other reactions to patients. And then eventually, of course, we have to develop a clinical protocol and consent forms and discuss with patients and get to the point where all of this is ready for a phase 1 trial. So once all of this is done and approved by the FDA, only then we are able to start the phase 1 trial. So I'll walk you through some of the work that we have done here for the RP-only transplant and the next steps of it today. So first step, how do you make RP cells from iPS cells, right? And this process is true for making any cell type. It's true for making photoreceptors, muscle cells, neurons, you name it. But essentially what you have to do is start with iPS cells. Your goal is to get to mature tissue. You essentially have to walk your iPS cells through various developmental stages. And the way we do that is you have a list of names here, and don't worry about the details, TGF, WNT, FGF, what they are. These are essentially growth factors and cytokines that we can buy from companies. And we add them into our cells to gently coax them into different steps, into the next step of the development, how they would have normally formed in an embryo. And we know this knowledge from all the research that has been done for decades and decades on people trying to understand how a human body is made. And this has been done in animal models, in mouse, in rats, in chicks, and various different animal models. And we have collated all of that information into one protocol and trying to make RP cells with a very high efficiency. Our goal is to make pure RP cells, because now we don't want to make the entire body. We want to make just the cells that we want to transplant, right? So that makes it even more harder to try and avoid not making anything else, because these cells have the property of making everything. So we have to make sure that these cells just stay on their path towards making RPE. And at the bottom, I can show you here, we've been successful. On the left, you see a dish full of what is called the RPE cells. You see little black dots. These are black dots, you see them as because these RPE cells are pigmented. It's the same pigment that is present in our skin, this brown-black pigment. So that's why they look brownish black, and that is actually for us an indication that we made the right cells and they're pure. And then in the middle is an assay that we perform to demonstrate. It's called a flow assay. We look for markers of RP purity, and here's an example, more than 98, we're now at 99.5% purity of these cells, and that is the limit of detection of our assays. Another important point is reproducibility. We have to make it from multiple patients. We have to demonstrate this process works. It's not just a one-time fluke, right? So we've done it over 3 dozen different donors now, and every time we're able to make RP cells when starting from their iPS cells. And then comes a time then that we transfer this process into clean rooms. So like I said, the clean room has a much higher standard of manufacturing, of making sure particle counts are very, very low. Again, details are not important, but what is important to understand is this is a long, complex process. It takes about 6.5 months. From the day we start draw blood from the patient, we reprogram their iPS cells, reprogram their blood cells into iPS cells, and then take those iPS cells and make them into RPE, and then make the RPE into a tissue. It takes time. But what we had to do, and we've done it successfully, is map out every single step of the process, every single day, what is going to be done for these 6 months, and how the cells are going to be slowly coaxed into RPE cells. And at the same time, for us, it's important to demonstrate that we have quality controls criteria. We're just now we're not just randomly making one or the other cell type. We have quality control criteria at every step of the process to ensure that we are going in the right direction. Our cells are sterile. That's the most important thing, that we're not adding any new bugs from outside, bacteria, viruses, COVID. These days we have had to add additional tests for COVID. Cells are normal genomically. They continue to belong to the patient. We didn't mix them up. And then as they're differentiating into the RP fate, we again ensure that they are becoming RP and they're pure RP. We look for purity. And then once they become RPE, we look for, again, sterility, purity, identity, and demonstrate function, that these cells are indeed functional before we transplant them. So very complex process. It took almost 3-4 years for us to optimize this protocol, but really we have been able to do it. And now there are many labs around the world who are starting to do this work. So to transplant these cells in the eye, we want to make a tissue out of it, right? So we just don't want to squirt cells in the eye. We want to make them into a tissue and transplant the tissue so that it starts functioning. It hits the ground running as soon as it goes in the eye, so it starts functioning right away. To make a tissue, what we do is we artificially put them on a membrane outside in a dish, and that membrane is made of a biodegradable substance called PLGA. And this substance, as you can see, has this little fibers about 400, 500 nanometer, and they're fused such that cells grow on top of it. And as cells continue to grow on it and mature on it, they make their own membrane. And this biodegradable material eventually just degrades away. So now it's a natural looking tissue because they're natural cells from patients' own iPS cells, and they have made their own natural matrix, their own membrane. So this gives the tissue much higher fighting chance to integrate in the back of the eye without any artificial thing added to it from outside. And I'll show you how beautifully it integrates. So this is how then, as I said, we have to characterize our cells. We spend a lot of time making sure that we made the right cell type. These cells are hexagonal in nature. So that's one way for us to look at that. These are the right cells. We look for the markers in green and red, 2 different markers we have looked at, and that light up their borders. And by the location of these markers, we know that we have made the right cells. We look at these cells at a much, much higher resolution sometimes. And this is one example of that, where we're looking at these cells using an instrument called a scanning electron microscope that many of you have heard of. So this instrument can look at subcellular resolution. And what you're seeing here actually is our transplant, which has been pseudo-colored. In blue is a scaffold. In brown, you see 3 cells, so you're seeing it from a side angle. And if you remember those hair-like structures, I told you in the beginning in the schematic I showed you, these are the hair-like structures that we have now recreated in these cells in a dish. They are shown here in this green color. And I hope you can appreciate. So this would be like 1 cell, 2nd cell, 3rd cells. Each bump, as you see these bumps, each bump is essentially a cell. So you can see about, I would say, 30, 40 cells in this tissue. I hope you can appreciate how homogeneously formed these hair-like structures are on top of these cells. And really, I think that's really for us the most fundamental proof that we have made cells. And we've compared this data to, let's say, work done on animal eyes and even cadaver human eyes. We know that these cells look almost identical to what is present in the back of the eye. So then once I showed you that, we know we can make the right cell type, we want to be testing them in animals and show that they do work, they are safe, and they do work at least in animals before we can transplant them in patients. So here's an example of transplanting our RP patch in a rat. So in this case, the rat we took is genetically immunocompromised, so it will not reject human cells. And human cells are here labeled in a red dye. You can see this red color labels the human cells. And what you see here is a choroid, rat choroid that I'm pointing to. And then the rat RPE is in the corner here, and this is a human RPE. And see how beautifully the entire tissue integrates into the back of the eye. The scaffold that we used to make it, it's no longer there. It has its own membrane that helped it nicely integrate into the back of the eye and even interact with the rat RP cells here. So this really gave us confidence that this tissue will work perfectly once transplanted in the eye. And this kind of work led us to do a very large rat study with about 450 rats that we transplanted with these cells. And we analyzed them at 4 different time points. Again, details are relevant, but what we were looking for is any signs of toxicity. So we analyzed these rats for the blood, urine, food, body weight, all things that you can imagine. And then we look for signs of toxicity, or if the cells are going away from the eye, crawling out of the eye elsewhere, or if the cells are becoming tumorigenic. And I'm really happy to report that even after doing 450 rats, we never saw any signs of toxicity or biodistribution into wrong organs or tumorigenicity of the cells. So these cells are really safe, and they're safe because these cells are fully differentiated into the final tissue, right? So these cells are really functioning as the normal tissue of the eye, and they just go in and they see, oh, that's an eye, that's in our atmosphere, how we should be working here. So they start working as they should have been.
Dr.
So then comes,
Kapil, can I, oops, excuse me, may I ask a question? We had somebody from the audience ask a question. It was from Kathy. It said, can you speak about the potential use of AI and robotics in the iPSC differentiation as a means of streamlining the production of these replacement RPE and photoreceptors? And if that's done now or later, that's fine.
Yeah, that's an excellent point, Kathy. Both are being done. I actually took out the slide for AI just in the interest of time. So we have used AI. Let me go back, and actually I can show you what we did. So as I said, these cells are hexagonal, right? And and as they become more and more closer to natural RP, the hexagonality becomes more and more perfect. And I also said they are pigmented. So as they become more and more mature, they also attain that pigment. So, and we realized at some point that that feature is extractable by AI. So we wrote an AI code, and we trained the code on the hexagonality and pigmentation to really predict are the cells fully mature or not. And it was just beautiful that after just training one set of we had 10,000 images that we trained on, it started seeing any new cells that it had never seen before, and would tell exactly what the maturity level of those cells was. So the AI, and we are using that feature actually in our clean room now routinely to confirm that what we made are the right RP cells. So AI is being worked on routinely. Robotic is something that we are working on right now. I'm working with actually 2 different companies. One of them I'm really excited about, that is using a closed system combining AI and robotic to make iPS cells. And it's a pipeline of system that will start with the blood. Everything would stay closed again to keep it sterile. And at the end of the pipeline, in about a month and a half or 2 months, we'll have iPS cells. This will take away manual subjective work that we still have to do sometimes. It will reduce the cost, and it will make the process much more efficient. So it's coming up. There's a company that is based in Boston, Celino Biotech, formed by 2 very talented women who did their PhDs in MIT. They reached out to me late last year and asked for my guidance on this. So I've been working very closely with them to make this happen. And I'm confident in the next couple of years, we'll have iPS cells made with robotics. So now comes an extreme challenge that we are now currently working on. I'll show you how we're trying to address this, is how do we demonstrate that this RP patch would potentially work for choroideremia patients, right? So we have to create an animal model in which we see similar damages to the back of the eye that is seen in choroideremia patient eyes, right? So for instance, here's an example of OCT. I'm sure many of you are familiar with optical coherence tomography. Is a technique that is used to look at the back of the eye by reflectance of light. And in this case, what you see here is one patient at different time points, age 37, 43, and 53, how the retina of that patient was changing in those 10, 15 years, and how these arrows point to changes that is happening in the retina. And those changes are reminiscent of the schematic that I showed earlier, where the cells are dying off, the photoreceptors are dying off, and because the retina, the RP has been not functioning properly. And in fact, I may have, yeah, so here's a hard mag here. I'll come to that in a minute. So actually, maybe we should talk about that now. So if you look at the lower left corner, again, optical coherence tomography, OCT of a choroideremia patient retina. And this yellow arrowhead points to, this is a very interesting spot. So this is where the RPE is still functional, or is present, actually, not functional, I would say. And you see this, it's very hard to see because these are very, the images are not high quality, and this is, signal is very, very tiny. But what you see from the, if you can follow my cursor, to the right is RPE layer that is present. To the left is the RPE layer that is now missing, because cells are dying. And now right above that area, you see these photoreceptors, these clumps of photoreceptors, they're rounding up, they're dying. And that is a very typical feature. So since we don't have a good large animal model for choroideremia at this point, we're trying to recreate these features using a laser depletion of the RP. So now what you come on the top here is a schematic of what we call a micropulse laser reduced RP ablation. So what we do is we use a specific laser of a wavelength which is only absorbed by the pigment of RP cells, which is shown here on the right. And RP cells would die off, very similar to what you would see in choroideremia patients. And then as a result, photoreceptors would die off. So now what you see is at the bottom on the right side, I'm comparing the OCT to a OCT from a pig eye, which has been laser damaged. And again, this yellow arrowhead points to the point where RP stops. There is a functional RP, or RP is present. Retina above looks healthy, but when the RP is gone, look at the retina, it looks different. You know, it has all this what we call hyperfluorescent spots when the photoreceptors are dying and clumping up. So really, so it's not exactly the same, but it's a situation similar to that we have created. What happens in choroideremia eyes is by using laser damage, we remove the RPE. As a result, photoreceptors start to die. And the idea would be now we transplant a new RP, new human iPS-derived RP here, and have it protect the big photoreceptors to demonstrate that our human RPE transplant is actually working. Another important feature that happens in choroideremia eyes is degeneration of the choroid. That's why disease is called choroideremia, right? So this is, again, a very difficult image to read, but what you see here on the left side, follow my cursor, you see these black and white speckles. Essentially, each white speckle is a choriocapillary taken in a Z section. And so it's very dense, right? Because that's how they bring very high confluency blood nutrients from the blood into the eye. But if you look into a choroideremia patient, these orange arrowheads point to areas where the choroid is dying. And you can see, you can compare the confluency of the two. The patient choroid looks much less confluent, and the areas where there's many vessels are missing. And now imagine in these areas, the retina is not getting any nutrients. So that's a big problem with the choroideremia eyes. And we have tried to recreate a similar situation into porcine eye, again with the laser damage, as we remove the RPE. And again, this white areas are highly confluent, normal, healthy looking choriocapillaries. But here, two different spots where we did this square size damage, laser damage to the RPE. You see the difference? All the choriocapillaries are gone. You only see the big vessels. So all of these areas, the retina is now not getting enough nutrients. In fact, we can tailor it by using different size of different power of laser. If you use high power versus low power, we see different amount of damage to the choroid. So this is in a way of us trying to recreate what is happening in patients' eyes into these pig eyes by using laser damage. So now we can transplant RP patch and have it protect both the choroid and the photoreceptors. So this is the model. Again, I'll come back to this. We want to go into the areas where RP cells are missing and transplant an RP patch that I showed you we can make on a biodegradable scaffold with patients on stem cells and have it protect the photoreceptors above and the choroid below. So to transplant a patch, we have to develop an instrument that can deliver it to the back of the eye. So we designed an instrument that is, here's a whole design of the instrument that is controlled through actually a hydraulic system through a foot paddle, so surgeon can maneuver the patch. If you go into the high magnification of the tip of the tool where the patch sits, this is how we would see. The patch is like a band-aid, you can see here in the middle. And then the tip has this very unique curvature that, as you know, eye is curved at the back, so it really fits that curvature of the back of the eye, and had to design this tainted green color so that surgeon could actually view the tool once it was in the eye, but could also see the patch, so it could really maneuver their way into the right place. Now, in the next slide, I'm going to show you a video of how the surgery is done in pigs. So some of you who may not want to see the video can skip this slide, just giving a warning here. So eye has been prepped for surgery. As you can see, 4 ports have been put in. Surgeon is now making the hole in the eye wall bigger. And after the hole is big enough, they will put the tool in the next shot. Here's the tool. And the retina has already been prepped, so skip that part. Retina has been cut. Now you can see the RP patch, pigmented cells on top. As you can see here, what surgeon is trying to do is align the tip of the tool to the cut in the retina. And once it's aligned, it will deliver the patch right under the retina. And then the retina is flattened on top of the patch, so it stays in one place, right? So now, and again, we can use techniques like OCT to demonstrate that the patch is delivered at the right place. So here's on the left, you see from the front of the eye, we can see the patch. This was a cut in the retina. And then again, the side view that I've shown you before. This yellow line points to where the patch is, and you can see this very bright straight line, that is a patch. And it's hard to say in this magnification, but we have experts telling us that the retina over the area of the patch looks healthier as compared to retina further away from the patch, which has been laser damaged. But we have other ways to functionally demonstrate that. And we do that by using a technique called multifocal ERG, multifocal electroretinogram of the eye. And this is done because retina is an electrically conductive tissue. So when you shine light, the electrical signals generated by the neurons that go to the brain. You can measure those electrical signals, right? So we can measure those electrical signals by specific instruments, and the one that we use called multifocal ERG. We shine light onto the healthy part of the retina. We get this nice and big response. You see this big curve here. When we shine light to the laser part of the retina, which would be somewhere here, we don't get any response because the retina is dying. Signals are low. When we shine light onto the transplanted part, which would be here, which was laser damaged and transplanted, we see a signal that is in between the healthy and the injured area, suggesting that human RP patch is indeed able to protect pig photoreceptors from dying, pig retina from dying, and hence we can measure its electrical response. Once the animal is euthanized, we can also actually confirm this by histology. What I'll show you is 2 different areas from the same eye, with laser area with no transplant and laser area with the transplant, and the transplant is marked here with the yellow line. What you want to do is compare the thickness of this cell layer, that is the layer of photoreceptors. These 2 arrowheads point to that thickness of the photoreceptor layer above the transplant as compared to away from the transplant, and it's almost double in this area, right? So as this is because the human RP patch is not letting these photoreceptors from dying in the laser area, whereas in the absence of any RPE, these photoreceptors continue to die. So again, for us, it's a proof that human RP cells are functioning to protect the photoreceptors. That's their job. That's what we hope they do, and in fact, they continue to do that. So I think I almost towards the end want to show you that the disease, I started off with saying that diseases affects all 3 layers, photoreceptors, RP, and choroid. And there may be cases when transplanting RP is not sufficient because the photoreceptors are also gone. So we may have to also transplant photoreceptors into those eyes. And to do that, we are now actually making a dual photoreceptor RP transplant. And this is the work we're doing with David Gamp's lab. And I heard he gave a talk, I guess, a few weeks ago, so you may have heard about some of this work. But essentially what they're doing is a similar approach to what we have done, is taking a scaffold, and in that scaffold, they're growing photoreceptors. Again, these different green, yellow, red dots are showing different types of photoreceptors that they seed in those transplants, in those scaffolds, excuse me. What we are now trying to do is make a sandwich, if you will, of RPE and the photoreceptor transplants. So here what you see on the left side is our RPE transplant, on top is a photoreceptor transplant. We combine them and we transplant them together into the same pig eye, same laser type pig eye. And what you see here is on the right, again, OCT, extreme right corner, healthy looking retina. You can tell the difference from healthy to the laser part, right? It's all the bright spots where the retina is dying, as compared to healthy, there's no bright spots. Now compare this to this part where the two yellow arrows point to, where we had dual transplant of RPE and photoreceptors. And you can see the transplants, in fact, with these two flat lines. The retina over this area looks almost identical to the healthy retina. So this dual transplant of RPE and photoreceptors works even better, we think, in protecting the pig photoreceptor or pig retina from dying after the laser damage. So this work is still at early stages. We're not at a stage that we have gotten the FDA approval for this part. We only have the FDA approval for the laser, for the RPE only part. But it's really, really interesting for us, because it's telling us that we are able to also restore the photoreceptors by doing dual transplant. So what we're now doing is using a technique called adaptive optics, which can look at the back of the eye at a single cell resolution. So now, same OCT that I just showed you, at the bottom you see adaptive optics data. So start with the healthy here on the right side, the white arrow points to the healthy retina, and you see each red dot is a cone photoreceptors. And you can see nice array of cone photoreceptors in the healthy part. Now, if you go to the laser part, green arrow points to the laser part, you see much fewer red dots that we have counted here. And that's what we expect, because we remove the RPE, photoreceptors die, we see less of them. Now come to the part where we have dual RPE photoreceptor transplant. We have much higher number of photoreceptors in this area. We can't tell the difference whether these are pig photoreceptors or human photoreceptors, but at least we know that we can restore the photoreceptor density once we transplant them in the eye. So this work is really exciting for us, because it's really addresses the late stages of diseases when there's no other hope left. We think we can restore the retina by dual transplants. So with that, I want to thank the team. This is perhaps my most important slide. As you can imagine, I don't do any of these things. I have this wonderful, wonderful team of people I've been really fortunate to work with, and it's a very broad team. This kind of work requires people with very different areas of expertise, from stem cells to developmental biology, physiology, surgeons, and people who doesn't know how to work with animals. And I've been really fortunate to work with all of these people, and actually for their efforts, what they have accomplished so far. The team got an award from the NIH director, Dr. Collins, who is standing right here. So this is all of us together receiving the award. So with that, I'll end my talk, and I'll take if there are any more questions. Thank you very much for your attention.
This was great. Thank you so much. I'm going to leave my video off because you might want to reference another one of your slides, Dr. Bharti. The first question comes from Keith, and it's a good one. He said, I'm confused a little bit. Don't these induced stem cells still have the gene defect?
Yes, yeah, they do. And I actually skipped that part for a reason, because I was hoping that we can discuss that in the discussion. So thank you, Keith, for bringing that up. Yeah, so I think what we'll have to do is take, and in fact, this has been done, proof of concept of this has already been done. When we start, let's say, for a choroideremia patient, start with their blood cells, and when we make the iPS cells, we will have to do gene therapy at the iPS cell stage. And it's doable because, as you know, gene therapy, if we can do in vivo in the eye, it's going to be much easier to do ex vivo in the cells. So we'll do the gene therapy at this stage, and then we'll make them into RPE cells. I know of several groups that have done this for other indication, not choroideremia, but for other similar indications. So it's a process that is doable. But yeah, you're absolutely right. We will need to do that.
Okay, here's another question from Brent. So the dead blood vessels at the choroid would be regrown or replaced by receiving stem cell treatment? Or is this treatment limited to affecting only the remaining, oops, I gotta go ups, only the remaining living blood vessels?
So what we know is that RP cells are able to help blood vessels regrow in the choroid. So we are hoping that, let's say, we put the transplant in one area where the blood vessels are missing, but if there are blood vessels in their joining area still alive, cells will secrete enough cytokines to recruit the blood vessels underneath and help the blood vessels grow. However, we are working on making a choroid as well that I didn't show because that work is also very early on. So we could potentially also transplant a choroid if needed.
Okay. Do you have any ideas about what vision would look like? And then secondly, is the patch, or would the patch be centrally located, or would it include the periphery?
So I think, as you know, this is a disease that starts in the mid-periphery. So one would imagine it would be better to transplant in the mid-periphery. And but that's again to be, discussed with the surgeons how to best do it. But I think that would be the right approach in this case. And I think once it's transplanted, one would expect that the vision is regained initially, of course, or like I said, the part, let me go back to my slides perhaps. All right. So if we can see this slide here, one would expect that these peripheries would start to come back in terms of their visual power, and then the night vision would come better as well.
I think that answers the question. Okay, we have several more. This is from Eric. Is the transplant being approached from within the vitreous to be placed below the choroid, or is the transplant approach FSU-RA, back of the eye, and put under the choroid?
So a good point, Eric. So currently the RP transplant is being approached through the vitreous. We remove part of the vitreous and go through the retina, under the retina, above the choroid. But if we were to also do a choroid transplant, we may have to go through the choroid for that. And we're working on strategies, how best to address that. That's an additional challenge that we have to work on, yeah. But the RP and the RP plus 4 receptors can be done through the vitreous, much easier to do. And we have already optimized that procedure.
Okay. Now, Brent asked a question. Either in the second or the third slide, it showed a 12 to 18 year timeline from early research to a commercially available product. Where in the timeline do you feel you are at currently?
So we are at what is called the phase 1 study, at least for AMD. For choroideremia, we're still at the preclinical stage. We're still at this point. But I should say that all the work that I showed you today that is going into phase 1 for AMD, all of that is applicable to the choroideremia part. Now, we have already demonstrated we can make the tissue, we can transplant it in animals. So we have done all of the translational work. What we need to do is paperwork for the FDA to get approval to do this kind of work as a phase 1 trial for choroideremia. So the timeline for that doesn't have to be 5 to 8 years. It is going to be much shorter once we start working on that.
We always try to pin down our researchers. We always like those timelines, and we've learned from most of our researchers that's hard to do. It's a hard question to ask. It doesn't ever come quick enough,
Yes. No, I understand that.
Marion asks a question. If the recipient for this procedure has from birth stored his umbilical cord blood, would this be used, or could this correction be used? Or actually, I'll add on to Marion's question, for child planning, family planning purposes, would it be useful to keep cord blood?
I would say at this stage, there's no need to do cord blood storage. The reason is, like I said, we can make iPS cells even from your adult blood or skin or fibroblast, right? So at this stage, the technology is advanced enough that all of this, the storage of umbilical or cord blood is not required. However, if you've done it, it's much easier to make iPS cells from the cord blood. They are actually much healthier because, as you know, the reason why stored cord blood is because they already at the progenitor stage. So they already halfway through. So it's easier to push them full into the iPS cell stage. But again, like Keith asked, we will still need to do gene correction in those iPS cells, right?
That makes sense. Do you want to touch upon Neil Bench, the president of the CRF, had a quick question, and he's asking if you would just touch upon the need or idea of having a CHM pig model, the value of having that.
Yeah, yeah, that's a good point. We've been discussing that with Jess for a while. As I showed that, we are challenging, it's a big challenge for us to recreate some of those similar anatomical changes that is happening in the choroidemia eye using our laser model. So this slide shows you that, we come close, but it's not exactly the same. Now that leaves us with a caveat that if we test our transplant in these conditions, which in fact we already have tested and we know it works, that the caveat is that whether the laser model is applicable to things seen in a choroidemia eye. And because in laser we laser ablate, and within 2 days we quote unquote go to advanced stage disease. We don't have that progressive stages that one would like to see in a choroidemia eye. So we can't pinpoint what is the best stage to transplant the RP patch or the RP4 receptor patch in a choroidemia eye based on the laser model. That is a challenge, because I think the ultimate effectiveness of this will depend on the right stage that we will find for transplantation. And for that reason, I think the porcine model would be highly, highly valuable.
That's good to know. I think that's a great thing. And we'll be talking about that a little bit this weekend, and I'll share that with everybody here after we're done. More questions are coming in. Thank you so much for taking the time, sir. Here's another one from Carol. Since stem cell therapies have been approved by the FDA for certain cancers, does this benefit the approval process for other diseases?
I think so. Let me just first clarify that the stem cell therapies that are used for cancer are a completely different class than what we are trying to make here. So for instance, let's see, what's a good idea? So maybe this one. So those cells are actually in the third category. So those are essentially blood stem cells, perhaps even mature blood cells that they isolate from patients, genetically modify them to kill the cancer, and then put them back into patients. So that's the category that has been approved. There has not been a single stem cell therapy made from ES cells or iPS cells that has been approved to make a replacement tissue. There's one company that has gotten approval to do trials for iPS cell derived blood cells to fight cancer, but not as a replacement tissue. In fact, I think in the United States, our trial is the first phase 1 trial for iPS derived replacement cell tissue. So it's a long way to go. But yes, more and more FDA learns about stem cells and stem cell derived therapies, it's going to be easier for them to approve any new therapies that come along the way. Just to give you an idea, for us to get this approval, the IND document that we submitted was 12,430 pages long.
Oh
so
my goodness.
That's how much regulatory input FDA requires when something new goes in. But as now they have learned from us that there are things that they can skip from the next people coming in, I'm sure they will be giving approval much sooner and with fewer pages.
Well, the next question kind of goes back to that from Paul. Subject to approvals, when would you think a phase 1 human trial could possibly take place?
For choroidemia, I can't honestly give you a timeline. The part of the reason is that we need some funding to get to that point, right? And this is work that we've been discussing with many people, with Dave and Dave Gam, and also members of the foundation, we need somebody to sponsor the trial to do the IND studies to get to phase 1 trial. We are also applying for internal funding. I was just told that the grant was in the funding range that we applied for, but we haven't gotten the money yet. So this will all depend on how soon we can get enough funding to get to that stage.
And just to give our members who are listening today and our guests, you know, this is something that having you a part of our team, Dr. Bharti, is great, because we would, you know, be able to work closely with you to see if there's any way that we can participate in that funding, because of course this is something really important to us. So
Yeah.
just to assure our members that are here today, we have a pulse on this, and we're going to be working real close with Dr. Bharti as it continues to develop, for sure. All right, I got to keep rolling. Go ahead, sir.
Sorry, I just wanted to add there that, you know, really input from all of you is really important here, because I've been talking to, let's say, companies like Spark Therapeutics and asking them that they should sponsor such studies. And having you also push for the same thing would really help get it perhaps approved by them, right?
I have people texting me and chatting me, asking questions, so I got to roll through these. That's good, though. I think you're right. It definitely demonstrates, it certainly demonstrates the merit of having this kind of relationship with a small organization such as ours, but it's definitely patient-driven, which is key, right? Okay, here we go. We have another question from Brent. It says, depending on the size of a macular hole, we do have people in our organization with CHM who have macular holes. So this is a good question. Depending on the size, could the 2 millimeter by 2 millimeter scaffold both bridge the hole as well as treat CHM, or is that two separate treatments?
So it will definitely bridge the hole, because holes are often actually less than a millimeter. They're very rare that they're bigger than a millimeter. But often the hole can also have loss of other retinal cell types. So for that, we will need the photoreceptor patch as well, at least. So yes, the RP only would bridge definitely, because it's bigger than that, bigger than any hole size that would happen in the macula. But we will need the dual patch to really treat it.
Excellent. You know, Eric has a question. You mentioned using gene therapy to correct the induced pluripotent stem cells. Can a gene editing be also done through CRISPR?
Yes, easily. Both of them are possible. It will depend which of them is easily available to us for, let's say, clean room manufacturing. Yeah.
Paul is asking some really great questions. Are other countries undertaking similar research to complement the work that you're doing here?
Yeah, the RP patch is being tested. Let's say right now, there's one study that is in London ongoing. There's one in California that has been ongoing, not different country. And then there's one in Japan that's been ongoing. So they have, and sorry, one in France as well. So yes, so I know of 5-6 different studies. All of them have similar approach. Most of them use embryonic stem cells, not iPS cells. The one only in Japan had used iPS cells to make RPE. That one is the closest to the way we are doing it, yes.
Let's see. Is your procedure expected to work, still work in late stage patients with very minimal vision?
Depends. If the photoreceptor layer is gone, then no, because the RP only will not be able to restore photoreceptors that are dead. But photoreceptor loss is sequential. It happens after the RP loss, and as I showed, there's a thick layer of photoreceptors, so they're slowly starting to die. So if some of them are still alive, we have sufficient evidence from animals that our RP patch will be able to revive the dying photoreceptors. But if they're completely gone, we will need the photoreceptor patch as well. And I should also, actually, going back to the gene therapy, the 2 gene therapy questions, I should mention that there's another approach to go after this, where we, rather than making the patch from patients' own stem cells, we make it from a healthy person's stem cells, who doesn't have the choroideremia mutation, and then transplant them as allo transplant. In that case, patients may need immunosuppression, but there are approaches people are trying to work around using immunosuppression. So there's another way to go after this. We may not have to need gene therapy. Gene therapy will be required only in the case of autologous transplant, which is made from patients' own stem cells.
Only two left. It says, let me see. Kathy's asking if you are in touch with Dr. Stone in Iowa, who's working to tackle the challenge of scaling the production of large numbers of IPS so that many patients can be treated. Are you familiar?
Yeah, with actually it's Bud Tucker, work very closely with him as a close friend. They are also trying to do autologous cell therapies for photoreceptor mutations that affect mostly retinitis, different types of retinitis pigmentosa. And he's also trying to develop an automated process. So I think, what I like to say is that you shouldn't have just one, iron to heat up. You should have multiple iron rods to heat up, or multiple ships to fire, because who knows who will eventually be successful. And we can always learn from each other in these cases.
Wonderful. And, I think you answered this question, but she did a follow-up question, Dr. Bharti. How will this individualized complex treatment process be streamlined so that many patients can benefit?
Yes, I think this is going back to what Kathy had asked. It's using things like robotics and artificial intelligence, where we really develop a streamlined process from patients' cells, making closed systems to make iPS cells, and then perhaps closed systems to do CRISPR-mediated gene correction, and then closed system to make the eye tissue. And then once it becomes a robotic process, ideally we can make hundreds of tissues in parallel.
You know what? Here's another one. So did I understand correctly that late stage could be solved if a photoreceptor patch was used as well?
Yes, so it depends again, what do you mean by late stage, right? So as I said, and well, it's a progressive disease. There's initially RP loss, and then perhaps as a result, there's photoreceptor loss. So if we find that sweet spot, the Goldilocks spot, we only transplant the RP before the photoreceptors are gone. We may not need the photoreceptor transplant, but if you're past that stage where both RP and photoreceptors are gone, you may need both tissues. And that's why I think, like I said before, the pig model would help us really nail that down.
Well, I think this presentation certainly gave our organization and members and our leadership team just great things to strive for. Certainly using money that our members have generated to continue to work towards a viable treatment, especially for our members. Gene therapy has given a lot of hope to our earlier onset. Again, nothing has been approved yet, but that certainly is something that, we're hoping proves to be valuable. But again, for our members and for our CHM family who are more advanced, we're really focusing as an organization to try to find a viable treatment as well. So this is very encouraging. And until that time, Dr. Bharti, here, I'll put mine here too. Until that time, would you suggest, for our members who are more advanced, anything that we can do to keep our eyes healthy, or that we need to do to just prepare for this?
I'm not a clinician, so that's out of my pay grade, above my pay grade, I would say, to suggest anything for clinically that can be done at this point. Sure. But I think, just keep the hopes up and keep fighting. We all are in this together.
Well, that's great. As your shirt says, right?
Yes, as my shirt says.
That's right. And we just want to say one more plug, guys, for everybody who's still on board with us. On Saturday afternoon at 2:00 Eastern, we're going to be doing a State of the Foundation, which is an update for where the foundation is right now in terms of research. Jess Thompson is the chair of our research committee, and he's going to be updating what projects we're currently funding and our hopes for the future, as well as internal things within the foundation. So if you can come, it will be recorded, but if you can join us, we'll be having a lot of people addressing the group. If you have nothing else to do, if you don't have a puppy job or something, you can come to Dr. Bharti.
Thank you.
But again, thank you so much. Thank you for joining us. And you were really able to take a very complex situation or complex research and be able to show it to us easily to understand. So thank you.
My pleasure.
You bet.
All right,
Happy
everyone.
Holidays. Thank you,
too.
everybody. And once again, just want to make sure we say thank you to our sponsors, Allergan and Biogen. And thanks everybody for joining us today. Have a great day.